Thanks very much, and I know these are very brief presentations, so I'll crack on. So the largest screen I've ever presented to anyway, so I hope everybody can see it. In fact, you'll probably want to read this next slide, the disclaimer in detail, since it's readable. I'm just going to give you a snapshot of the company in such a short time. We are quite a large company, and one of the many challenges is actually just people talked about earlier in some of the sessions about how difficult it is, how slowly trials are going. They also forget that actually behind the scenes, you're also growing the company itself and actually having to build up all of the teams. We actually now have 80 people in Philadelphia and about 200 in Didcot, just outside Oxford in the UK. And to put that in context, we were beginning of 2012, I think we were 7 people. So this is you have to build a company at the same time as develop the processes, as well as obviously pushing forward the clinical trials. So we're entirely focused on T-cell receptors. This is it's similar to a CAR T-cell in the sense that it's autologous T-cell therapy. But what's very different about it is the targets. It's different in lots of ways. First of all, they can't be recognized by antibodies, and therefore they can't be recognized by CAR T cells because they're not whole proteins on the cell surface. But secondly, as I said earlier, for those of you here, there are actually lots of these targets. So we've been looking, we usually are looking at around 60 targets at any one time. And because of that, you can find lots of targets which are closely connected with cancer and therefore not on healthy tissue. And in particular, there's a class of antigen called the cancer testis antigens, and these tend to be absent in normal, healthy adult tissue. So we have lots of targets, and we are otherwise very similar to a CAR T-cell company in the sense of autologous T-cell therapy. So there are lots of things you've got to get right, though, with a T-cell receptor. First of all, it's target identification. I know that sounds very easy. Why shouldn't you know what the target is? After all, these are well-known targets at the protein level. But if I tell you there are at least 3 companies now with TCRs in the clinic around the world where we absolutely have shown that the peptide is not on any cancer cell. And these are companies running trials right now. The other issue is the TCR identification, which is very, very important. We've been at this since I founded a company called Avidex in 1999, and what we've learned over the years is that not all T-cell receptors are equal. Some are cross-reactive, some can't be engineered, and they are very, very different in their performance. And I think this is something very much overlooked by the industry. You really need to get a TCR at the right affinity and one without cross-reactivity to have a successful program. That's why we've invested heavily in the engineering. We have a lot of patents around how to engineer the affinity. Once you do all that, though, you also need to look at safety testing. It's very, very important. Cell therapy, including ourselves 5 years ago with a program, had some deaths, and cell therapy in general, I think every company has had deaths, and the academic institutions. So safety testing for each TCR for us takes over a year now. But we are looking very much to the improvements, Generation 2 and Generation 3, things that will increase the durability, in particular, of response and reduce the ability of the cancer to fight back against the T-cell system. And we put all that together, and we call them SPEARs, for specific peptide-enhanced affinity receptors. As I said, we have 4 key components. We have the right targets. We only select low-risk ones for our programs because we have so many. We can guarantee the right affinity. We've got a special way of making TCRs now without using T-cell clones, which are notoriously inefficient at producing engineerable T-cell receptors. So we use a completely different technology, and that means the attrition has almost disappeared in the affinity maturation of the T-cell receptors. I've talked about the safety testing. And actually, I'm not going to go into it today. We've heard a lot about manufacturing. We have spent a long time, and we have a complete process development group engaged in manufacture. But I will say one thing, and that's that the manufacturing using the lentivirus and the beads, the CD3, CD28 beads, which we license exclusively from Thermo Fisher, we license them for all T-cell receptors. Novartis licensed them for CAR T-cells. And if you look at the persistence of cells in man, it's several years for us and for Novartis and not for anybody else. So we think that manufacturing is critical, not just in cost of goods terms, but in producing the right type of cell to have good persistence. We've got lots of trials going, as it says here. It's really the pipeline for an industry, not a company. We've got trials running. These are the target antigen names. So NYESO originally stood for New York esophageal, but actually is present in a number of different cancers. We've got studies running or complete in 5 cancers. That's under option to GSK. We have INDs open for MAGE-A10 at the AFP, and we'll file the IND in the first quarter next year for MAGE-A4, which is probably the most prevalent cancer testis antigen across solid tumours of all of them. And then behind that, we're looking at a series of targets. And as it says here, we drew up a list of the top 20 cancers that we wanted to look at, and we've actually got targets for 19 of the 20, and generally speaking, we've got 2 or 3. So what are the studies we've been going? So one of the speakers earlier said that many of the cell therapy companies don't do large trials, and there's a reason for that. But actually, we're trying to work out exactly what is going on in each cohort, and we're doing that in the trials. But secondly, it is my belief that if you don't get a signal within a cohort of, let's say, 10 patients, it's not going to be a product anyway. These cell therapies are not going to be used if they're marginally efficacious in 1 in 50 patients. So if you don't see a signal in a relatively small cohort, then I don't think it's really a therapy in future. So we finished cohort 1. That was using preconditioning of cytoxan and fludarabine, the optimum, very high doses actually, and also people who expressed MYEs at a very high level. So in other words, optimum conditions. So what we got was a fantastic response. We got six responses, 5 partial and 1 complete response in that. We then are enrolling a study looking at, well, does the level of expression of MYESO matter? That's one of the criteria. So we're looking at deliberately low expressors. We then took out cohort 3, we took out fludarabine, and in common with others, actually, when you take out fludarabine from the preconditioning, you lose efficacy. And it's to do with the immediate expansion of the cells after the infusion. If you don't get that expansion, then you don't get much efficacy, and if you don't use fludarabine, you don't seem to do that. So we're now just started. We announced, I think, a couple of weeks ago that we started cohort 4, which is looking at the same group of people as in cohort 1, but with a modified, much reduced version of the preconditioning. So what we're doing is we're going stepwise, trying to work out exactly what are the parameters that really matter in order to maximize the efficacy. So, and just to illustrate the sheer potency of a T cell, these are pictures before and after. These are 2 different patients, obviously. The top one, as you could see, a great shrinkage of the tumor, but more important, the metastases disappear, so these patients become eligible for resection. The bottom one is just below the knee joint, and you can see again, this was actually within 2 months, I think the top one was within 6 months, that you get this decline and resolution of the tumour so they can be excised. And that's obviously, although these are partial responses, both of these patients actually were tumour-free after resection. We also did a study which we published on multiple myeloma. This was in the context of autologous stem cell transplant. Again, we got great data. We got a 91% overall response rate at day 100, which is the typical day measurement for multiple myeloma. We published 3-year overall survival at the beginning of this year. And we also got a 59% complete response rate in those patients. The next thing here, though, multiple myelomas are tricky because it's a very it's an attractive disease and there are lots of new medicines. So what we signed last week actually was a deal with Merck to look at a combination deal with Keytruda. So that's what we'll be doing next. We'll be doing a 2-arm study, one with Keytruda and the cells, and one with just the cells, and we'll be doing a comparative. That will be a sort of randomized study. Again, only a couple of dozen patients. We are running an ovarian cancer study with 10 patients. We didn't get any responses without fludarabine, and so we've just put fludarabine back into the equation, and that study has started. Melanoma, again, we started with 4 patients, no fludarabine, we getting no responses. And we are just recruiting in non-small cell lung cancer as well, and that's with fludarabine. We haven't released any data on that. So there's a common feature, as I've stressed here, and that's that if you take out fludarabine, whether in sarcoma, you don't have it in ovarian, you don't have it in melanoma, in each case you don't get the responses. And in both synovial sarcoma and in an earlier study in melanoma, we had seen responses. So we do think this preconditioning now has been very much, is very important, and that we've sorted it out. So we have our own programs, as I say. The next one where the IND is open is MAGE-A10. It's another one of these cancer testis antigens. It's on different diseases. We've started a trial in non-small cell lung cancer, and we've just announced that we are doing a triple study in bladder, melanoma, and head and neck cancers, and that's actually going to be our first collaborative study with our new strategic partner, the MD Anderson Center in Texas, where we announced a deal with them about 2 or 3 weeks ago. AFP is an unusual one for us. AFP stands for alpha-fetoprotein. The IND is open. We don't have vector right at the moment, so we won't start dosing till next year. But AFP is unusual because it's not a cancer-testis antigen, and also it's only for a single cancer, liver cancer, so we should get some data next year on that. And then behind that, as I mentioned, MAGE-A4, which is another cancer-testis antigen. So when I say we have lots of targets, we really have lots of targets. The IND behind MAGE-A4 will probably be around the end of next year, probably 2 each a year after that, if that's the route we want to go with multiple targets. And that's shown here. We have a full pipeline where at the very early stage, we're running 11 targets, where target validation, 6 targets, and we can run a proper discovery program for 6 targets in parallel at any one time, and that's what we're able to do. And as you can see, they address multiple diseases. Although, despite my accent and despite the fact we were formed out of Oxford University originally, we're extremely American in our outlook, and in fact all our clinical trials, bar one centre here in the UK in Manchester, have been carried out in the United States. We've only ever dosed 2 patients outside the USA. This is a list of the sort of great and good hospitals in the United States, particularly in cell therapy, and we are working in all of these centres. At the beginning of this year, it was 9 centres. It's now over 20. And these are the things that are very important to establish these relationships with the major cancer centers in the United States if you want to recruit patients and deliver data. We've had an extraordinarily productive year this year. These are sort of long list of achievements this year, but starting actually with another branch of the empire, which we expanded into autoimmune, the concept we use Tregs instead of killer T cells to dampen down the activity of the T cell system. We expanded our collaboration with GSK. We got breakthrough status and orphan drug status for NY-ESO. We also got prime status in Europe for NY-ESO as well. We filed 2 INDs. We got the AFP filed earlier this year. And recently, I think probably the ones at the end of the list have been really very important to us. Without wishing to blow Bob Pretty's trumpet at all, we did a 5-year strategic deal with PCT to make these. That's very important to secure the manufacture of these cell products. We do all of our manufacturing in Allendale, New Jersey, under PCT, and we're building a pilot plant ourselves. We did the multi-year strategic alliance with MD Anderson, just announced a couple of weeks ago. We've just started that cohorts 4 in synovial sarcoma, and we've also just started the ovarian cancer. So we're going to be producing lots of data next year, and we will also start the study with Keytruda as a combination, our first ever combination study with a T-cell therapy in 2017. We've just signed that. So it's been an incredibly active year, and you can actually see when people talk about what's actually needed, you have to focus on everything from manufacture to combination therapies to preconditioning regimens and do it all at once. And that's obviously what we're focusing on, and delivering data in patients in 2017 is critical to us. We will have a lot of milestones coming up. We'll be filing the IND for MAGE-A4 shortly, so in the first quarter of 2017. We've actually should be able to announce the next T-cell target during 2017. And actually next week we're presenting the first preclinical data, SITC, Society of Immunotherapy and Cancer in National Harbour, the first preclinical data of our first second-generation molecule. And what these are designed to do is to counter the hostile tumour microenvironment of a solid tumour. So you can actually show that you can still retain killing in vitro, even in the presence of the microenvironment that you'd find within a tumour. So we're presenting data actually at CTOS in Lisbon next week, and at SITC on both that and actually all the preclinical data on MAGE-A4, from which you will see it's pretty highly expressed in a lot of cancers and very specific to the cancers and not on normal tissue. We should start a lot of studies during the next year as well. We've got a study in myxoid round cell liposarcoma to accompany the synovial sarcoma. That should start around the end of this year. The AFP, as I say, when the vector is ready, we'll do that. The combination study in multiple myeloma, which I think will be very interesting to see if there's synergistic or additive effects of adding a checkpoint inhibitor. And lastly, we should start our synovial sarcoma pivotal study during the course of next year. Obviously, we've been talking to the FDA about the design of that study for some time, since we got breakthrough status early this year. And so we should have a design that is satisfactory to the regulatory authorities in the first half of next year, and start that around the middle of the next year. So there'll be a lot of data coming out in all sorts of things, NY-ESO in lung cancer, ovarian cancer, and in MAGE-A10 we should also have lung cancer, and data from those first 3 triple cancer study running at the MD Anderson Center. In addition, I hope we'll have safety data from our next 2 programs. So that was very much a whistle-stop tour, and I better stop there. I'm probably just about on time.